I'm Charles Wondji, and I'll be talking about the molecular drivers of insecticide resistance in malaria vectors. It's important to start by mentioning that vector control really plays a major role in the prevention of most vector-borne diseases. And this is done usually through long-lasting insecticidal nets, LINs, and indoor residual spraying, where insecticides are sprayed on the walls or live residing. And at the bottom here, I just want to ensure that vector control have really played a major role in reducing the burden of malaria since the year 2000. And as you can see on your right, the by 70% to the reduction in malaria cases worldwide. But we are facing a major challenge because there is a growing and widespread report of insecticide resistance in most malaria vectors. As you can see, this is the situation before 2004, where if you look at the African continent, more dots were green, meaning that vectors were susceptible. But back in 2015, what we have is more red dots showing really widespread resistance. And on your right, I put a picture there that I took myself in Uganda, where mosquitoes, fully blood-fed, were actually resting inside the bed nets supposed to kill them. Why? Because of resistance. What we need to know is that, unfortunately, there are only limited insecticide classes that could be used for the control of public health pests. We have 4. Among them, organochlorines with DDT, organophosphate. One example is Temefos. We have carbamate. One example here is Propoxol, but we also have bendalcap. We have pyrethroid with type 1 pyrethroid. One example is permethrin. Type 2 example, deltamethrin. And it's good to mention here that pyrethroids are the most used for control program. And the reason is they have the lowest toxicity to humans, and that's why most bed nets actually contains pyrethroid. All these insecticides, what they do is to target the insect nervous system. And if you take the case of DDT and pyrethroid, they both bind to the sodium channel. And as you can see on your right, the model displayed there, what they do is that they keep the channel in an open state, and what happens is that they cause repetitive firing of neurons, leading to paralysis and death of the insects. For organophosphates and carbamates, they rather bind to the acetylcholinesterase, and they probably produce the same effect, also leading to death of the insects. I just put there a model of acetylcholinesterase docked with an insecticides. So what is the genetic basis of resistance? What we need to know is that often the wild population is susceptible before the insecticide is applied. But as we apply the insecticides, the few resistant that were in that population will be selected, and the proportion will increase naturally. And the resistant insect, therefore, will be at an advantage, and they will reproduce, and the genetic changes that confer resistance will be transferred from parents to offspring, and resistant insects will become more and more predominant. As you can see in the cartoon here, as we apply insecticides, the proportion of resistant allele increases significantly. And this will even go quickly if the allele is dominant, and slower if the allele is recessive. And if the resistance is actually dominant, as it has been seen in the population, in a very short number of generations, we can move from susceptible to resistant population. Another question is, what are the sources selecting resistance in the field? One of these is agricultural practices. Why? Because the same insecticides used in agricultural sector is also used in public health. And that makes that insecticide used by farmers actually contaminate vectors, breeding sites, and that will induce resistance even before these insecticides are even used in the public health sector. And that's why there needs to be a strong collaboration between agricultural sector and the public health sector to improve the management of those existing and few insecticide classes. Now, why should we bother about resistance in public health? Does it really matter? I think I will say yes, but just showing this slide where through a modeling, WHO predicted, for example, that if we are not careful managing resistance, that may lead to a significant increased mortality of up to 260,000 deaths a year if nothing is done. So it's not just an academic issue, it's really something that can impact public health seriously. Now, let's talk about mechanism of resistance. We have overall 4 main mechanisms. We have what we call the target site, where the target of the insecticide is modified, preventing insects to bind, and therefore it will not produce the lethal effect expected. We also have behavioral resistance, when mosquitoes change the behavior to avoid exposure to insecticides. We have what we call reduced penetration. As you can see there, the insect will thicken the cuticle, limiting the amount of insecticide that may go in, and therefore mosquito will be able to survive. We also, and one of the major mechanisms is what we call metabolic resistance. And in this case, resistant insects are able to produce large amount of enzymes that are able to break down the insecticide before it reaches the target site. So these are the 4 main mechanisms of resistance. Let's start with the biochemical resistance mechanisms. We focus on these 2: target site, metabolic resistance. For target site, we have various types. This one is what we call the knockdown resistance mechanism. And this type is actually caused by a single amino acid change that occur in the gene called sodium channel transmembrane protein. And that leads to the KDR type of resistance, knockdown resistance. These mutations are actually able to confer resistance to pyrethroid and DDT resistance. Leucine to phenylalanine substitutions is really something widespread concerning this mutation. But we have to bear in mind that not all species have this type of mechanism. So it's not always expected that all mosquitoes will express the mechanism. For this, there are simple DNA-based diagnostics tools allowing us to actually easily detect the resistance in the field. The other target site is called acetylcholinesterase. And in this case, we have a glycine which is replaced by a serine at position 119 of the gene, and confer resistance to organophosphates and carbamate. And again, not all species of mosquitoes will have it, because other species like Anopheles phoenaceus will have a different amino acid change, asparagine to isoleucine at codon 485. So we always need to bear in mind that things are not standard. Now let's look at the metabolic resistance, which is one of the major ones, as I mentioned, because it's more likely to lead to operational control failure. That's why we pay attention to this mechanism significantly. And it's also a bit more complex because it's not contrary to target site. You don't have one enzyme involved. You have, for example, 3 major enzyme families involved besides small ones. And the major ones are cytochrome P450 monooxygenases, glutathione S-transferases, esterases. And the reason why this mechanism is complex because there are many enzymes potentially involved. We are talking about maybe 200 enzymes. And the challenge is to know which one is really driving resistance. And even if you find the one that is associated with resistance, the other challenge is that there are other mechanisms through which that single gene may confer resistance. That may occur by a point mutation in the coding regions. It might be mutation in the regulatory loci or the transcription factors. So there are many mechanisms making this mechanism very complex. But what I would like to share with you now is that we have made significant progress in detecting this resistance mechanism, starting by detecting the major genes which are overexpressed when we talk about pyrethroid resistance. For example, the several genes, P450 genes like sub6P3, sub6M2 have been detected as playing a major role in Anopheles gambiae. This has been done using transcriptomic, using functional genomics to detect those genes. And also concerning other species like Anopheles spinosus, another major malaria vectors in Africa, using transcriptomics based on RNA-seq method. Major genes have been detected across the continent. But something interesting was that, you know, mosquitoes from different regions of Africa, they don't exhibit the same genes to be resistant. So making it even more complex, because what's happened in one, let's say in Central Africa, is not the same in Southern or East Africa. So that means that it's not one size fits all in terms of implementing control program. What about detecting the specific genetic variants which are conferring a metabolic resistance? Progress also has been made there. And in the slide we have here, I'm presenting the case of the first DNA-based metabolic resistance that we detected. And this was in a gene called glutathione S-transferase epsilon 2, where we detected a single amino acid change from leucine to phenylalanine at position 119, which was actually acting by enlarging the substrate binding pocket, allowing more insecticides to get in and to be metabolized. We show that this amino acid change was strongly linked with the phenotype, those having it able to survive more than those without it. Using that, we designed a simple DNA-based assay, allowing us to crack the spray of resistance, which was located mainly in West Central and East Africa, not in the Southern Africa. And using this expertise, we move forward to tackle the case of cytochrome P450, detecting also a specific polymorphisms which are tightly linked to the resistance phenotype. And we use that information to design simple DNA-based assay, allowing also to track resistance. What we did was to first of all confirm that the polymorphism was really correlating with resistance. And indeed, we show that once mosquito have that polymorphisms, it's able to survive exposure to pyrethroids. That was done for the first gene, but also for another P450 genes, confirming that yes, DNA-based markers for P450-based resistance could be detected in malaria vectors. And now that we had such tools, we could answer key operational questions like, what is actually the impact that metabolic resistance is having on the efficacy of bed nets? And in Cameroon, we built experimental huts, mimicking a situation where people are sleeping under the bed nets. We can release mosquitoes there with different genotypes and compare those that have survived exposure to bed nets, those that are blood fed, to find out what was the impact of the genotype. Overall, what we show through that experiment is that when mosquitoes have the homozygote resistance allele, are homozygote for the resistant allele, they are able to survive better when exposed to pyrethroid-only nets. They can blood feed more, meaning that it's reducing the efficacy of bed nets. But the good news was another type of bed nets that we call PBO nets, synergist net, because it incorporates a synergist which is able to block the activity of P450. Those nets are actually more efficient even when mosquitoes have the P450 resistant allele, which is showing that in the field, control programs should rather use those PBO nets. And because we have another markers, we could answer the question, what's happened when mosquitoes have actually not just one resistant allele, but two? what we show is that there is an additive effect. So if a mosquito is double homozygote resistant, then it will survive much better. Just look here, the double homozygote resistant, they are all alive, whereas the double susceptible tend to be the one dying the most, showing that yes, we need to act before mosquitoes are becoming very resistant. And talking about molecular drivers, there are other factors. Mosquitoes can also become resistant by having what we call increased copy number of certain resistant genes. This has been shown in Anopheles gambiae, where some pyrethroid genes, pyrethroid resistant genes are present in many copies, allowing mosquitoes to survive. And resistance also could come, I mentioned earlier, through cuticular resistance. Mosquito can thicken the cuticle to limit the amount of resistance of insecticides getting in. And progress has been made recently by detecting the genes which are associated, including 1p450, that can help produce cuticular hydrocarbons. But for these mechanisms, we still don't have a molecular marker yet. And to make things even more complex, in end of 2020, it was shown that another mechanisms confer resistance exists, and this is through sensory appendage proteins called SAP, which can confer resistance in other malaria vectors. So we always have to be open-minded in terms of what is the mechanism potentially involved in the field. Overall, when we know what are the specific genetic variants driving resistance, there are key operational questions that we can help answer. One of them is resistance really increasing malaria transmission? We did that by using the GST E2 allele. We actually genotype those mosquitoes infected versus non-infected. As a result, we show that the mosquito infected were mainly homozygote resistant, showing that yes, resistance actually may lead in some cases to increase malaria transmission. And that was further compounded by the fact that those resistant allele, those with the resistant allele were able to live longer. So meaning that could really increase malaria transmission. Another question was, what is the fitness cost? Because if you want to implement resistance management like rotation, we need to know whether there could be a restoration of resistance, of susceptibility. And indeed, for P450 allele, we show that there is a restoration of resistance. If you don't apply insecticides, the strain may become susceptible over time, which is a good news that if we stop using pyrethroid and switch to another one, in a few years' time, the population may become susceptible again, which is a good news. And we also can look back to see really that using scale-up of intervention actually is driving the selection of resistance. Look, for example, this is in Mozambique. Before bed nets were scaled up, most mosquitoes were susceptible SS, but 15 years later, they are all resistant, showing that bed net is actually driving resistance in the field. But the good news is we are having new tools based on new insecticides like Fludora fusion using neonicotinoids or new bed nets using chlorophenol. So we need to take advantage of the knowledge we have gathered to better manage these tools and to implement appropriate resistance management to avoid having to deal with population which are now completely resistant. We need to act when it's still resistance is still at the low frequency. And how can we do that? Using methods like mixture, rotation, and mosaic to actually limit the frequency of resistance. To summarize, yes, resistance is a challenge for vector control of malaria, and the metabolic resistance is playing a major role. But the good news is recently we detected new diagnostic tools, and this is now allowing us to do more in the field. And this is actually something that should help us to detect resistance and to manage it properly. So that is really where I will stop here, and thank you for listening.